Wow, what an amazing opportunity. I just want to take this chance to thank Nanopore and the organizers for allowing me to show some of the research that we've been doing down in Wellington. So today I'm going to be talking about a collaboration that we have set up with Wellington Regional Hospital, or our NUI laboratories. So what we have done is set up a trial run where we have brought in the MinION device into the microbiology lab within the hospital. And when patients develop a hospital-acquired infection, a sample will get taken. The DNA, bacterial sample will be cultivated and grown. We'll do a DNA extraction, run it through the nanopore, and then feed the reports back to the clinicians. So for those of you that aren't quite sure where New Zealand is, we are quite very, very far away, as the slide clearly says, so it's been quite the journey. New Zealand comprises of two main land masses, the North Island and the South Island. So Wellington, highlighted in red, is based on the North Island, and that is where we are based. And the point of this trial is mainly to benefit patients. So this is only a research project, and we're hoping to give the infection prevention and control team more precision so that they can make better informed decisions for patient care. Now, before I go down the route of some of the really cool things that we're able to show with nanopore sequencing, I thought talking about a social science project that my colleague Suzanne Manning has been leading might give you a bit of something different to talk about. So during the trial, we went in and we were speaking to some of the doctors and the microlab staff and asking what they found the real barriers were from using the nanopore device in their labs. And to our surprise, it wasn't so much the, oh, I don't really want to do this, or I don't really know what's going on. There was that hunger there and that drive to learn and upscale. The main barrier, and I'm really sorry for saying this at the 10-year anniversary of the MinION, was the space for it. And I don't mean that by the device. The device itself is amazing and ultra portable. What I mean is we had to buy a super fancy and expensive computer, which takes up a bench space, which you can't really move. So we have to kind of sacrifice that space. It is also worth it as well. There was some issues with the computational side of it, so lack of bioinformatics expertise. A lot of these people that work in the hospital don't have access to it, but we have been able to overcome that, and that's kind of where I come in and my employer ESR. So to give you an overview of how this works, so a patient is sick, they've acquired a hospital-acquired infection. We take their sample, whether that could be, for example, blood or urine. We culture the bacterial isolate, do a DNA extraction, and then run that through the nanopore sequencing device, so the MinION in this case. So this is where the pipeline kind of splits into two. So for the hospital, they are really concerned with a species ID and a multi-locus sequence type. And by multi-locus sequence type, or MLST, that's kind of a typing scheme where we take 7 conserved genes, and that's species dependent. And then depending on the nucleotide sequence of those genes, we can assign an allelic profile to them, and henceforth a sequence type. So once they have a species ID and a sequence type, that gives them information on whether or not there is an outbreak in a certain region, area of the hospital. Now, alongside that is where I come in, and that's where I kind of do, and to quote the clinician involved in this, the really nerdy stuff. So we transfer the sequence data to our group at ESR, and we use some Dorado-based calling and a lot more in-depth analysis to do comparative genomics and phylogenetics. All in all, those two streams come together for a report that gets fed to the clinicians and the infection prevention and control team, where ultimately they can make better decisions for their patient care. Now, I'm only going to talk about 2 examples today, given the time limit that I have. The first one I'm going to talk about is Staphylococcus aureus, Methicillin-resistant Staphylococcus aureus, or MRSA as it's more commonly referred to. So last year we had 2 neonates come into the Neonatal Intensive Care Unit with crusty eyes. So we took swabs and followed that workflow, and we were able to say that this was MRSA sequence type 97. So that was obviously a problem. You got 2 babies in the NICU with the same sequence type. So we opened an investigation and screened all the neonates in the unit, and pardon me. And we found another 9 babies with asymptomatic carriers of this MRSA sequence type 97. So the power in this was we were able to identify that outbreak very early, but from only 2 infections. The other 9 were asymptomatic, colonized. So 2 weeks after the last case of sequence type 97 was found, we were able to declare the outbreak closed. Now, this is one of the success stories, and hopefully the power of nanopore sequencing really comes through here. So back in 2007, another hospital in New Zealand had an outbreak of another Staphylococcus species, and unfortunately it kind of grew and grew and grew until December 2014. It was so out of control they had to actually close the NICU and relocate it to a refurbished facility. So as you can imagine, doing this has saved quite a lot of dollars and potential lives. So after that outbreak, between June and August 2023, I got a phone call one evening after my dinner, and we had another MRSA sequence type 97 in the same NICU. So didn't get much sleep that night, but really kind of showed the power of what we were doing. So we were able to get a report back to the clinicians, and I hope that this slide will show you the case that was detected in November in 2003 is quite genomically distinct from the cluster that we found earlier on in the year. So that was a huge relief for the staff, for the hospital, and more importantly for the parents. And we didn't have to go in and swab all the neonatal babies in the neonatal unit. So yeah, it's really, really powerful tool. And the second project that I'm going to talk about, the second bug I should say, so is about Clostridioides difficile, or more commonly known as C. diff infection. So between February and August 2022, we sequenced all the hospital-acquired C. difficile infections. So these graphs show the top 5 sequence types that we were able to detect. And one thing that kind of stuck out like a sore thumb was sequence type 2 on Ward B. So with that information, the infection prevention and control team went in and did some interventions. And by interventions, what I mean is we supplied gloves, some aprons, we increased staff awareness and education, and changed the chemicals they were using, the cleaning chemicals, to one that was more efficient against the C. difficile spores. It was a little bit delayed thanks to the COVID-19 pandemic, but I have indicated where the intervention took place, and as you can see, it kind of dropped off the face of the ward, so to speak. We did detect 2 more cases of sequence type 2 later on in the year, but then doing the retrospective phylogenetic analyses, again, I could show that those 2 cases weren't genomically linked to that original cluster. And if you want to know more about that, this research was published earlier in the month in Infection Control and Hospital Epidemiology. And then just to kind of finish, a picture says 1,000 words, and that is true in this case. So this is my collaborator, Dr. Max Bloomfield. He's the driver of leading the genomics within the hospital. He shows a really keen interest in using this technology and can really see the value. And it is a privilege that I can work alongside him to make his dream a reality. So much so, and Max has also been key in establishing the Genomic Special Interest Group across New Zealand, which has resulted in us establishing another base down in Dunedin Hospital using a similar setup. So when that finally gets up and running and we start generating data, it'll hopefully give us a lot more insights into other hospitals as well. And with that, you know, it's not just me and Max. There is an army behind us, a lot of people. I think I want to give a shout out to Miles Benton, who now works for Nanopore. This was his dream, and it's been an honor to try and show him what we're able to do. And of course, we are trying to go down the One Health approach. So just a shout out to all of our external collaborators at LIC, and of course, the diagnostic labs across New Zealand. Their hard work is what makes this a reality. And then thanks for Genomics Aotearoa and the Ministry of Health for providing funding for that for this work. Thank you.